We are going to talk to you today about what we perceive as the state of the art of DNA data storage. Before I do that, I want to quickly introduce you to DigNA. DigNA is a funding umbrella of the European Union. European Union has identified DNA data storage as a, let's say, important future technology, because in the future we're going to store much more information as we have to store today, and our traditional storage methods have real drawbacks that we can't store data for long times. Storage is relatively costly and generates a lot of waste, and there's been literature to show that DNA might be able to overcome some of these challenges. However, there are, let's say, technical and scientific challenges in getting there, and as this has been identified by the European Union, they have invested into funding research and development in this, which is this funding umbrella DigNA, with the vision to creating expertise within the European Union and become an expert group in enabling DNA as a data storage medium. This DigNA is organized as one part of the European Union. They're funding nine different consortia. Some of them are small, one research group. Some of them are relatively large with multiple research groups inside and companies. And in between, there's a steering committee which organizes this whole overlap. And as part of, let's say, the activities, general activities within this program, we have decided to create a webinar series. And this is, let's say, the first instance of this webinar series. And the idea of this webinar series is to give educational background from experts in the field about various aspects of DNA data storage technology. We are aware that this is an extremely interdisciplinary field and it needs a lot of different expertise. And throughout this series, we'll invite speakers from different aspects of these technologies to give interested people the required background. Today, we will start with, let's say, really a broader envelope to talk about the state of the art of what we perceive is the state of the art of DNA data storage, where we see that are some of the problems. And then if there are, let's say, more specific knowledge needed, that will be covered in, let's say, later lectures within this webinar. This webinar, for all of your information, is the ideas that we have talk about for 45 minutes between me and Tomaž. After that, that's a Q&A session. The webinar itself is being recorded. We'll turn off the recording for the Q&A session so that you can feel free to ask any questions and you don't have to be afraid that, we believe that that's a good way to run this, that we can feel freely, that you can feel very free to ask your questions. With that, I'll go directly into what we want to talk about, which is really what is today, or what do we perceive. And I have to say from the beginning, this is a biased view, because Tomaž and myself, we've been in different aspects of DNA data storage throughout the past, for me, nearly 10 years. And so this is what we perceive as, let's say, the state of the art. And of course, when we talk about the state of the art, we have to talk about, let's say, where do these ideas come from? Who created these ideas of DNA data storage? And for that, there's this very nice, let's say, interview of Norbert Wiener from 1964, where he states that magnetic tape as a memory in a computer will be replaced by genes. And he also says, I know it's very difficult to read, my hunch is, and I'm not alone in this, that the next decade or so, we will see this used technically. But this was already 60 years ago. It took quite a long time until we really technologically came to the state that we are at least much closer to really storing actual information in DNA and replacing a hard drive of a computer with something very similar to genes. And really the point here is that the ideas and concepts around this are nearly as old as the discovery of DNA. As soon as mankind understood how DNA works in our body, there was very quickly the idea, well, if biology stores information that way, why don't we go ahead and store digital information in that way? The problem was more that the tools to, let's say, interact with DNA were missing. So how would you read and write? And so where these ideas existed in the 60s, the first experimental implementations I know about is this one on the left from Joe Davis. He's interestingly an artist, working, let's say, in the biotechnological space for many years. And he wanted to store this microvenus, so this symbol that you see on the bottom of the left in DNA, and together with collaborators, as far as I know, at Harvard, he then created coding on how he would, let's say, store that in DNA. And at the time, it was possible to, let's say, chemically synthesize these short DNA sequences that you see there, which encode, let's say, this primitive image. And as the text says, yeah, so that the microvenus was chemically synthesized, the DNA, and then embedded into a vector, and this vector was then embedded into bacteria. So there was, let's say, this idea of living information. But also at the time, or in this, let's say, scope, this was not, as far as I know, never reread. So the information was in these bacteria, and this was, let's say, first way of writing. But to my knowledge, there's no reading in that. So in a, let's say, similar time, a little later, let's say, more, let's say, technical scientific paper, Cleland in Nature, had the idea of hiding information in DNA. So writing in this case, they could write more DNA. So they wrote June 6th, invasion Normandy. So back to, remember, D-Day. And then had these oligos synthesized, and they hid these oligos in a large background of, let's say, non-specific DNA as a, let's say, steganographic means of hiding information. And they were not only able to synthesize much more DNA, but they could also read the DNA. At the bottom, you see the Sanger sequencing information, where, let's say, let's say, text was then sequenced, so first PCR amplified and then sequenced and recovered in that way. And for, let's say, our field, as I perceive it, a big step was in 2012 and 2013, when quite at the same time, Goldman and Church, two research groups, stored about 1 megabyte in DNA. I'll start with Goldman because they actually submitted their paper earlier than the others. It was published later, but it was submitted earlier. And they took text, image, audio, and code of about 740 kilobytes and stored that in DNA, in short DNA oligos. So the idea here is, in contrast to how biology stores information, that information is stored not in one long DNA molecule, because long DNA molecules are very difficult to synthesize, that the information is stored in many short oligos. And as you can see here, you see here overlapping. So the idea in this paper is that you have overlapping sequences of, let's say, short oligos, which store this information. And also similar at the same time, as I told you before, is this paper by Church in Science. Very similar amount of information. They also stored a book, images, and code, and were able to show and that, let's say, this is a very dense storage format. Also, the, let's say, messaging of the two papers are a little different. Goldman is really about, let's say, stability, let's say, long-term being able to, and the Church paper is really goes about density of, let's say, storing information in DNA. So I want to go a little more in detail how these two, what the technologies are that enable this work of storing 1 megabyte of data in DNA, and also to go through the process a little. So this is from the Goldman paper. you can see that this is the time it took them for the work. It took them 2 days to synthesize the DNA, which is actually also here done by a commercial company. Then they shipped it to Germany, and then it took them, I don't know, 15 days, nearly 15 days, 13 days to sequence, to read back the information, because at the time sequencing was still relatively slow. So it took to read a megabyte of information took 2 weeks. And this also gives an idea of, let's say, the processes involved in this DNA data storage. In, let's say, more conceptually, what we have to do if we want to store information in DNA, we first have to find a way how we translate zeros and ones to ACTNGs. Then this, let's say, list of ACTNGs have to be synthesized. This synthesized DNA is usually amplified so that we have enough of it, and so that we can purify it and get rid of, let's say, incomplete synthesized DNA. There's usually the DNA stored for a specific amount of time. There's going to be decay during the storage. The DNA is read back to sequences of A, C, T, and G. And in the end, we have to decode this, let's say, information that's present in A, C, T, and G, A, T, C, and G back into zeros and one again, and to have, let's say, back the digital information that we originally wrote. And so the key technologies in that are really synthesis and sequencing. So the writing of the information and the reading of the information in DNA. And what I'm going to talk here mainly, and that's also how these 2 papers work, is DNA storage by synthesis. So really, A, C, T, and G are actually chemically synthesized, one, let's say, letter after the other one. For non-chemists, that can be imagined a little like Lego. You have building blocks of A, C, T, and G, and in your preferred arrangement, you can put them one on top of the other. In reality, that's a little more complicated. So these is a cyclic process where on the surface, you have the beginning of your sequence, which has protection groups, and then you take in blue these building blocks, which are added to the surface, and thereby you can grow DNA strands to a specific length. So the really drawback of this technology is how long the DNA sequences can be made. As I told you before, we can't make very long. Let's say our genome is millions of base pairs long. Here, with this synthesis, we can do perhaps 200, nowadays 300 base pairs can be synthesized. And where traditionally, with this method, you can only make 1 DNA strand per reaction. And you're quite limited, because 1 DNA strand, 300 nucleotides long, that's maximum 600 bits, because theoretically per ACTNG, you can store 2 bits because you have twice the number of possibilities. So you're quite constrained with just having 1 oligo. But let's say motivated really by biomedical research, this, let's say, DNA synthesis over the last 20 or more years has been automatized and miniaturized. So that the synthesis doesn't happen on a large surface and the whole surface grows the same DNA sequence, but miniaturized surfaces, which can be chemically addressed, synthesize on every different spot on a surface a different oligo. So we can, let's say, on a surface, on a chip surface, about a square inch in size, we can synthesize thousands, hundreds, thousands of different oligos simultaneously. There are at the moment 3 technologies how this can be done, how the surface can be addressable so that one position on the surface synthesizes something else than another point on the surface. One way to do that is an inkjet printer, where with an inkjet print head, you bring the base you want to synthesize in a specific cycle step of your growth. You bring that with the inkjet head to a specific position on the surface. The second technology in the middle is that you tell the oligo on the surface by electrodes, which spot on the surface you allow to react with the next base. And the third way is that you use light to direct where the synthesis should happen on the surface. And all of these technologies are actually commercial. There are different suppliers of these technologies for inkjet printing that at the moment mainly Twist Bioscience, electrochemical synthesis, that's GenScript. There you can buy oligos from those. Light-directed synthesis is just I'm sorry. Light-directed synthesis is just being commercialized where you'll be able to purchase the device and not the oligos. As I said, we can do about 300, let's say these technologies can do about 300 nucleotides long, and these pools can have up to 8 million parallel reactions. So this is really massive parallel, really massive parallel synthesis of DNA, and therefore also enables us to store a lot of information. Because for every bit we write, or for every 2 bits we write in the end, because as I said, we have to do one chemical reaction, and there's no way in cheating for that. Every 2 bits needs one chemical reaction, and this technologies really enable that. Here you can see such a chip surface. This is from custom array, now GenScript. You have this, let's say, on a silicon wafer, small electrode chips, and on every of these chip surfaces, targeted by electrochemistry, a different oligo will be synthesized. So a different sequence of A, C, T, and G. And when the synthesis is complete, the oligos are cleaved from the chip. And what we receive if we do an order at one of these companies is a small flask of water, and that contains all of these oligos cleaved from the chip surface. So that's writing. Reading, most people know more about, because reading is much more, we know much more about reading DNA from let's say genomics, that nowadays in medicine, if we have genetic disease, our genome can be sequenced. Usually that's done by a technology called Illumina sequencing technology. I won't go into the detail. That's certainly going to be done in one of the later webinars. I think what's interesting to know, it's also a surface technology. So where individual oligos that are to be read are attached to a glass surface and then amplified on that surface, and optically, the sequence is then optically detected on the surface. And it's firstly a surface technology, and secondly, also a massively parallel technology. So here, readout is also done base by base along all oligos on the surface in parallel, and millions of oligos. Nowadays, I don't know exactly the number what Illumina offers as, let's say, platform. Millions and millions of multi-million oligos can be read in parallel. As I showed you in these 2 papers, for Goldman that took 14 days, that process. Nowadays it's more overnight. There are new devices which are even faster, and speed has increased. But also what's super fascinating about sequencing technology is how cost has reduced over the last, let's say, 20 years, where 20 years ago the cost was probably $1 billion to have one human genome sequence. So this is always cost per human genome. That's about a gigabyte roughly of digital information if we think about it in digital. And nowadays this cost has shrunk to about $1,000 to read that amount of information. And if you think about the paper of Goldman, and these 2 papers, Goldman and Church, they were 2013. There the cost of sequencing was 10 times higher, and also it took them 10 times longer to read the information as we do nowadays. And we can somehow assume that this trend will continue to some extent, and that reading technologies get cheaper and cheaper as we move along. So these 2 technologies, they really showed that if we combine this synthesis technology with state-of-the-art sequence technology, we can read and write a megabyte of information in DNA. The challenge for both was that retrieval was imperfect. So they both had some points in the DNA, in let's say the data, where they had to, let's say, do some fine work, and let's say it wasn't fully automatic, the recovery. And also it was still unclear how would you do multiple files, how would you put, let's say, more structure into the data that you can. And then subsequent work showed firstly that using error correction coding, there's also going to be a future webinar about that. Error correction coding is quite common in data storage technology. In all data storage technologies used in CDs, hard drives, SD cards, they all use error correction coding, which a technology that enables that if the hardware of storing bits creates errors, we can remove those errors by storing a little redundant information. There's some quite hard mathematics behind that, so I won't go into that. But what can be done is to take those methods developed for hard disks or CD and adapt them for DNA data storage. And if we do that, we can recover information perfectly, completely error-free, although our synthesis, storage, and sequencing technologies will induce errors. If we know those errors, we can put a little more redundant information, store that together with the original data, and then retrieve our information error-free. The next addition to making things more practical is random access. So random access meaning that if I have a hard disk and say I want file 17, that I only get file 17 for my hard disk. And in DNA, the idea there was shown in this paper for the first time, that the idea that you put, let's say, primer sequences to the end of the oligos, which address a specific file. And then we can use PCR with those two primer sequences of the file that we want to read and amplify all sequences that contain those primers from the oligo pool. As I said, we store information in, let's say, many different oligos at the same time. But using PCR and a specific pair of primers, we can, let's say, amplify a specific set of oligos from that pool. And then if we do a sequencing experiment, we only, or to a vast majority, sequence only those sequences that we have amplified. So that enables the selection of, let's say, files, and we don't have to read all of the information that so we have some, let's say, pre-selection of data that we can store, and then one can think about how does one address that, how large is the pool, how many files do we put together into such a PCR primer, how many oligos do we put together, and really generate a structure into that. And this whole, let's say, error correction coding, random access, synthesis, and sequencing were really put together by a research group between Microsoft and University of Washington. They showed in 2018 that you can store 200 megabytes of information in DNA. You can do error, if you do error correction and random access, this really becomes a practical and scalable technology. And they not only showed that you can do that to a larger extent, they also showed that you can read it with nanopore sequencing instead of with Illumina sequencing. So let's say enabling a more modern, or even more modern way of accessing the information. They also showed that you can build a device. So really not, let's say, usually if we do these kind of DNA data storage experiments, we have a whole lab and we have workers doing, let's say, pipetting. And so it's very unpractical. It's usually not how you imagine data storage device, which is a hard disk, usually a cable where you send your information in and you get your information back. And this group, Washington and Microsoft, they built this device you see here on the right. It's about small size of a small table that does everything. So it has a cable going in that feeds the information, it synthesizes information, it stores information as DNA, and it reads back the information using a nanopore sequencer, which is also on this table. and it does all of that and sends back the digital information. So it's really a device. And that's a big step, although the amount of information they read and wrote wasn't very large. I think they stored hello world with this device. But again, it's really important to show that it can be made into a practical actual device and not only, let's say, a lab experiment as we very often do in our everyday work. This is what I perceive as the state of the art of, let's say, DNA data storage by synthesis. 200 megabytes, they've now shown they can do about a gigabyte. Let's say there's random access, you can retrieve the information error-free. That's really doable with that. Another point we have to touch on, really important for me as I'm a chemist, is that DNA is not perceived as extremely stable from, let's say, archival discovery. But on the other side, if we work with it in the lab, we know that we usually have it in the fridge or freezer because we are afraid that it breaks. And the reason for, let's say, this paradox is that DNA is relatively vulnerable to hydrolytic damage. So it reacts with water. So let's say, if you have a long strand, every site has a specific probability of reacting with water. So the longer the DNA, the higher the probability of having a break in that DNA. And so if we want to store DNA for a long time, we have to prevent reaction with water. And there are 3 ways we can do that. We can either have it extremely cold, and so the reaction is slowed down, or we protect the DNA from the environment, either by putting it into, let's say, a container, or similar to the bone, we put some, let's say, inorganic, some solid material around it, and thereby protect it from the atmosphere. And with DNA, that's also what we do. We either put DNA into the freezer. There's a company who's also part of the DigNA portfolio, Imagen. They make small shells in which they put DNA and inert gas. So the DNA in there is really protected from hydrolysis with water and is extremely stable when in this data storage format. Another technology is that the DNA is encapsulated in tiny glass particles, and this is like a fossil. So the DNA is really within glass, and the glass protects the DNA molecules from the surrounding water or atmosphere, and this also enables relatively high stability of DNA. So perhaps 500 years, the shells much longer, many thousands of years, because there's absolutely no water in there. And in the cold, depends of course how cold you make it, DNA can also be phase stable. So all of this together, we can say DNA, we can store megabytes of data, high storage density, error-free recovery, is scalable, practical, we can store it for a very long time. And then the question, well, why are we still using hard disks if this is also fascinating? The problem is that the technology we use at the moment, very expensive, especially synthesis. So let's say DNA synthesis is extremely expensive per megabyte. Of course, per base pair or the oligo, it's extremely cheap. But if we store megabytes or gigabytes, that really sums up and it gets very expensive. Sequencing is also still much too expensive to enable data storage applications, so archival data storage applications, and both technologies are also too slow. So to solve those problems, we have 3 alternatives. We either make synthesis cheaper and faster. There are approaches working on that. So using, let's say, the DNA synthesis we're using at the moment has been developed for gene synthesis, which has to be perfect. So one approach there is to use, let's say, less perfect, faster, cheaper DNA synthesis, have more errors. But as I showed you, we can compensate for errors with error correction. So there's certainly a trade-off in that. There's at the moment a lot of work in enzymatic DNA synthesis. So not using chemistry, but enzymes to make faster and cheaper oligos using biochemistry. Secondly, we can state, well, if it's super expensive to make gigabytes or megabytes in DNA, why don't we look what applications we can do with the information we can already store in DNA? Because if we want to store 100 kilobytes, that's very feasible and relatively low cost. But then the question is, why would we want to do that? And the third is find alternative read-write modes than what we do. And within, let's say, DigNA portfolio, different consortia are looking at various of these problems, or let's say problems, futures in DNA data storage. And I'll quickly talk about the second and third and the, let's say, state of the art in those, or what, let's say, we are really interested in. And one is really find applications not needing. So where, and the question there is really, where can we use DNA or as a storage medium where it can solve problems that are not possible to solve with hard disks or CDs? What can we do with, how's DNA special? And DNA is special because it's extremely small. It's a molecule and not a device. We can make many copies of it, and we can implement it into liquids, polymers. We can put it into different kinds of materials. And so this is what we call DNA of things. So it has the idea of putting information into objects using DNA. Actually, in our case, using DNA that we first put into glass capsules, and then we put these glass capsules into objects. As for example, this 3D printed bunny, which contains the file from which it was printed within itself. So if this bunny, in the bunny are small glass particles, in the glass particles is DNA, and the DNA encodes for the file from which the bunny was printed. So you can take a small piece of the bunny, let's say break off a piece of the ear of the bunny, take out the DNA, sequence it, and make a copy of that bunny. Another application of that is, application showcase of that is a music album has been stored in DNA, encapsulated in glass particles, and mixed into a graffiti paint. So an artist can paint with music, so that what is painted really contains the information in the paint. Or similarly, also with paint, this year Germany celebrated its constitution, what's called the Grundgesetz, and to celebrate that, group of artists had the idea of encoding this German constitutional Grundgesetz in DNA and having that DNA part of the ink of the Süddeutsche Zeitung, which is a relatively large newspaper in Germany. And we actually did that. So DNA, so this text was synthesized in the DNA, put into the printing ink, and 400,000 newspapers using this ink were printed, and every one of those newspapers had thousands of millions of copies of that Grundgesetz in the ink as DNA. And what's really special here, you can't do that with any other storage format. You can't put a CD or hard disk, and that's really where we think is a big advantage of using DNA as a data storage format, because how distributable it is, how you can put it into objects, materials, inks, and thereby give objects digital content. And that's something we are really fascinated about. But I also wanted to talk, and if we go even smaller in amount of information, we can put just a barcode into an object. So instead of putting, let's say, Grundgesetz, we can just put a number, for example, into cotton. And this is something that has been commercialized. So putting DNA again into glass capsules, and the glass capsules into cotton. And when the DNA is in these glass capsules, the DNA survives all the processing steps which happen in making t-shirts or other textiles. And with this, the origin of a material can be barcoded in the actual t-shirt. So it's not in a label, but it's really the cotton itself which gets barcoded information imprinted. And very recently, actually this week, the spin-off company has announced that they're having a larger campaign with a large European textile manufacturer. So this is to really show applications. There's not only applications of DNA in gigabytes. DNA is also really powerful. Even if we just have a barcode, just a few bits of information, it can be useful due to its different format it has to traditional storage means. However, as I said, we should also look at other ways of reading and writing DNA. And especially if we say that DNA is so difficult to synthesize and biology is full of DNA, why don't we use the DNA that's already there to store information? But the problem with the information that's already there in biology, it already contains information. It's the biological information. So the idea of, let's say, these new DNA storage approaches is to use long existing, usually natural DNA strands and modify them. So imprint information in some smart way to encode for information in addition to the information that's already biologically there. So there's no interest in the sequence of ACTG that encodes for the biological information is not of interest. What's of interest is, let's say, a modification of the strand. And there are a few ideas in literature. One is to do molecular recoding. So within bacterial cells, to feed them with specific DNA strands, and the bacteria using CRISPR-Cas in relatively complicated, at least for me as a chemist, ways, adds those oligos to the end of the bacterial DNA, thereby modifying its own DNA and imprinting, in this case, digital information of images of a running horse or film of a running horse into the DNA of the bacteria. Another approach is these DNA punch cards. I don't know if who of you has ever worked with this. It's long before my time of working with computers, but that in the past is how how computers stored information. You had a card and information was stored just by punching holes into those cards. And you can think of doing exactly the same with DNA. You have a long DNA strand and you kind of punch holes in it. And the information you store is where you put, let's say, a hole into the DNA strand. And the hole in DNA strand means breaking the DNA strand. So let's say having the long strand and let's say nicking it, putting in, let's say, by enzymes at specific locations, nicking the DNA so that it fragments, and then reading the fragment lengths. And actually the information is encoded in the read lengths of these fragments that are recovered in this idea. And it really directly resembles a punch hole card. Another way, also using existing DNA, is DNA origami, where existing DNA is assembled to, let's say, a flat surface, and this flat surface then has specific locations where other DNA or fluorophores or other molecules or particles can be attached to, and then one can read either by AFM imaging or by fluorescence the location, or if, let's say, specific rands have been, information has been added there, 1 or 0 has been added to that or not. And the most recent example is to take a long DNA strand and induce epigenetic modifications to the DNA strand. So in biology, epigenetics is, let's say, information that's in the DNA, but not as A, C, T, and G, but as other chemical, let's say, modifications of the DNA strand, often methylation of the DNA strand. And here the idea is that we take a long DNA strand and methylate specific locations of that, and the information is stored if either methylation is present or non-present. And these methylations can be done at specific locations with enzymatic reactions. And with that, many kilobytes of information can be stored. Let's say, however, at the moment, not to the scale that can be done with DNA by synthesis, where we can do multi-megabytes fully automated on these chip surfaces that I showed you during the synthesis. Here, automation is still, let's say, much slower, and things are usually in well plates and not automated to the same way as having 8 million, let's say, reaction sites at the same time. And with this, I'll pass over to Tomaž, because he's going to talk about other ideas, let's say, making DNA storage more practical, faster, cheaper. And yeah, I'll stop sharing and leave Tomaž to share his slides.
Hello. Hello. I hope you see my slides. Yes. Okay. So again, thank you very much, Robert, for a very nice introduction today. I hope I will be my part of presentation will be up to the level you presented in the past couple of minutes. So Robert has mentioned a couple of times that DNA synthesis can be tricky, slow, expensive, and so on. So there are also other methods, and one that's been well tested in the past is what is called a block-by-block synthesis, also known as array synthesis. The very basic idea is shown here on a slide. So instead of recording bit by bit, we actually try to record larger chunks of data. So essentially, as it's, again, this is a very simplified example. If we want to record a word hello into DNA, we map that sequence of characters into a pre-designed DNA fragments that are selected from a larger library, and then eventually assemble them together in a larger, longer DNA chain. Of course, this looks like very trivial here on the slide, but in reality, there are many challenges to achieve that result. So, showing again a very similar slide to what has already been shown, the block-by-block approach has similar steps as DNA-based synthesis. So essentially, we first convert zeros and ones into the desired sequences of SEGTs. We prepare the DNA by mixing the short fragments together, perform the DNA assembly, and in this way, the data is written to the DNA. The data is then stored in one way or another, and the reading of the data is again done first by sequencing and then by data decoding to get back to the zeros and ones. What's maybe important to stress here is that all part here, starting from the DNA synthesis here at the beginning up to the end of the DNA sequencing, relies on actual material flow. So the data, as we know it, is only at the very beginning when we input the data to this DNA synthesis process, and when we process the data that we get out of the sequencing process. Similarly, as for all other methods, the challenges are simply cost, making the writing process fast, and do the reading in an efficient way. Now, of course, we want to record more than a hello world. So essentially, we are dealing with large amount of data. So the first step, the DNA synthesis, to perform the DNA synthesis, we split the data into multiple parts, such as shown here, and then we prepend or a sequence number to each one, and then encode each short segment in a separate reaction. Then we simply run the assembly reactions, and eventually we can join all the output in a common DNA archive. Again, this looks very simple, but the issue here is that this second step here, the splitting the data into multiple parts and then combining them correctly into multiple reactions, this requires something that is known as a combinatorial explosion, because we have to choose from many different fragments in order to make the larger DNA chain reliably contain the data. So for the now that was more of a mechanical process where we have to mix different components together, while the DNA assembly is much like biochemistry. The emphasis of that part is more on biochemistry. So the idea is here to join those short fragments together, and there are typically two main options. One is the polymerase chain reaction, and the other one is ligase reaction. Again, there are a number of challenges here. We need to achieve high specificity. In other ways, we need to ensure that the DNA segments are assembled in the correct order. We need to achieve high yield. So from whatever input gets into the reaction, we need to, the efficiency there must be very high and no side products created. Number three, because we perform many reactions similarly to what was explained in DNA by synthesis, there are many reactions in a very small amount, in a very small volume, on a very small surface. So we need to avoid cross-contamination because that damages or skews or corrupts the data recorded. The reaction volumes are an additional constraint. If you want to make the synthesis cheap, repeatable, and something you can offer eventually to a customer, the cost of synthesis is of paramount value, paramount criteria. So reduction of reaction volumes is there the key element to make the DNA assembly efficient and cost effective. What it means in numbers. Typically, in a chemistry lab, the volumes are in order of microliters. So here, the reaction volumes, in order to make this process efficient, is a nanoliter or even much below that one. Last, but not the least, in order to make the DNA synthesis something that you can do on a regular basis, not just once in a while when you want to publish results, when you want to make some public statement of how good or how fantastic the particular method or approach is, we need to create a streamlined and high throughput and also parallelized operation. So all these combined together, these are the main challenges in DNA assembly. And of course, not all of them are solved, because if they were, this will be already something that would be offered on a daily basis. Now, after the data is recorded in the DNA, of course, we also need to read the data back. Unless you can do that, then then that makes no sense to record the data in the first place. So when talking about reading the data, in particular, when relying on sequencing of the DNA, this is essentially, from an engineering or mathematical theory point of view, this is a sampling process. So we are dealing with probability, statistics, and randomness. On the other hand, there is also a minimum requirement depth of sequencing. So it means that each data segment or each data carrying segment of the DNA needs to be read back several times to make sure that the read was correct, was done in a correct way. Robert has mentioned errors, and this is a fact of a technological fact. We can't work around it. But of course, there also be the signal needs to be about the noise. So we need to get more correct reads from the false or noise background. So combined, this limits the size of the DNA archive that can be read back. Now, it's very difficult to put a specific size of the archive we can read, because this number depends on so many parameters that are all listed here on the slide. So, for example, anything like from efficiency of the decoding software, encoding scheme, how well was the DNA assembly performed, what is the uniformity of the multiplication, and so on. So many parameters. So without the progress has been made, this is still a considerable limitation in the DNA storage technology. Lastly, but it is our experience that custom sequence analysis software greatly improves the process. The reasoning is actually very simple. When using block by block approach to DNA synthesis, the DNA has certain specific structure, which general sequence analysis software is not aware of. So if you do your own software, you can build in that knowledge into that software. So you can get the more data, more efficient readback through this custom designed software where this knowledge about the DNA structure is actually built in and also relied on. Now, there are pros and cons, of course, in block by block approach. First is there seems to be a higher data throughput during the writing process. It seems to be that there is a better match for streamlined writing operations. So it means something you can continuously write without any kind of interruption. Clearly, a major drawback of the block by block approach is lower data density. But on the other hand, this is compensated by the fact that in this way, there is a built-in error correction and prevention, at least at the very basic chemistry level. Think of it in this way: there is more several bases recorded or formed in the DNA that cover, let's say, 1 bit. So even if one of those bases gets wrong through the reading process, you can still decode what was the value of that particular bit. So whether it was 0 and 1, to make it again a simplified explanation. And there seems to be a lower cost. It's difficult to put a specific number here because it depends on what is the technology and how it is used. But again, similarly as what Robert explained, is still very expensive for the time being. The biosafety and biosecurity seem to be a little bit more manageable because, again, the structure of the DNA is pretty much known in advance, and you can't really get any kind of random DNA sequences that may pose a threat from a biosafety or biosecurity point of view. Now, the fact is that the error correction at data level, the one that Robert has explained, that must still be used, though probably the redundancy introduced at that level, of course, can be lower because some of that is already included in this block by block approach inherently. Again, this way, this is also truly multidisciplinary activity. So if you want to make this work, you need to have people understanding everything from chemistry to mathematics and everything in between. Now, how well block by block performs. Fact is that there is limited information available, and the most outspoken so far was Catalog, an MIT spin-off. And from what was publicly announced, they recorded 16 gigabytes of Wikipedia back 4 years ago, but there is no info whether that was actually verified or read back, at least not to my knowledge. In the same statement, they also claimed that they have achieved 180 gigabyte capacity per archive. This is my assuming here, and also that they can scale up to 1 megabyte per second throughput. Again, this was claimed 4 years ago. Now, from applications point of view, we think there are two main aspects of the DNA that really stand out. In addition to what Robert has explained, I think the medium obsolescence is the most important criteria here, because if you use the sequencing as the basis for reading the data back, essentially, as long as humanity is around, that the sequencers will be around because of the medical of the applications in medicine. So unlike the CD, if you had recorded one 20 years ago, nowadays you would have a hard time finding a CD drive to put your disk into to read your data back. This is really not an issue for the DNA. Another aspect is superior data security. As Robert has explained, it's almost a hidden, transparent. The DNA can be hidden everywhere. So the actual applications to to my view, need to be found somewhere in the intersection of the two aspects presented here. Last but not the least, think also of a James Bond application, because here, what we call it internally, because the DNA can be simply and easily disposed of. So, you know, burn after reading approach can work here as well. Yeah, and finally, last but not the least, if you have infinite amount of time and money, we can the technology is ripe enough to store actually the whole world's data in just one bottle of DNA. Again, the caveat is with infinite amount of time and money. And last but not the least, we are part of the DigNA portfolio with 6 projects. And again, we appreciate the funding we received from the EIC and European Union. So thank you very much for your attention.
Thanks very much.